Hello students, today we'll talk about one of the most fascinating experiments that paved the way for finding out the molecular basis of inheritance. You have already studied how Mendelian factors were proved to be the unit for inheritance, but what it was. The search began with a famous experiment by Griffith. Griffith postulated that bacteria is able to transfer its genetic material by the process of transformation. But then too, there were certain areas that he could not answer. Let us have a look at this beautiful experiment. So in 1928, Frederick Griffith conducted an experiment and he suggested bacteria are capable of transferring genetic information through a process known as transformation. Let us look at his experimental organism. Griffith used mice and he used Streptococcus pneumoniae, a bacteria that caused pneumonia. There were two strains available, one strain having smooth polysaccharide coat. It was capable of causing severe pneumonia, so that resulted in death of the mice, hence it was called as virulent strain. The other strain without that coat was not able to cause pneumonia and the mice was alive. So he conducted series of experiments rather say, with Streptococcus pneumonia, the bacteria that causes pneumonia. The two strains, as I have already mentioned, the two strands were type 3S smooth strain or S strain and type 2R rough strain, right? Two strains, rough and smooth. So polysaccharide capsule present in this case. So how does it help? Actually, because of its polysaccharide capsule, the host immune system is not able to destroy this strain. And as a result, it causes pneumonia and host dies. So S strain is lethal or virulent. The polysaccharide capsule is absent in R strain, hence it is defeated by the host immune system. As a result, the host survives. You know, there is a defense mechanism in every organism. So R strain is a virulent or non-virulent strain. In his experiment, he injected the R strain into mice and it was seen that mice was alive. Mice did not die. So what can be the reason? Because it did not have that polysaccharide coat and hence it was defeated by the immune system of the mice. Once he injected S strain inside the mice, it causes pneumonia and mice died. Now, the next important thing, he heat killed the S strain and then he injected the same in the mice. Mice was able to survive. So what is the inference? Heat killed S strain that causes mice to survive. The R strain also causes mice to survive, but he was surprised when he mixed the heat killed S strain and live R strain. Remember, both were not able to kill the mice, but when he injected the mixture, the mice died. This was something that he did not expect it. So what was the conclusion? When mice was injected with S strain live, mice died. Conclusion, S strain is virulent. When mice was injected by R strain live, mice survives. Conclusion, R strain is non-virulent. When mice is injected with S strain heat killed, mice survive. Conclusion, virulence is destroyed on heating. Now, the most important aspect. Mice was when injected with S strain heat killed and R strain live. Mice dies. So what conclusion do you draw out of it? The conclusion is live R strain and live S strain were recovered from the dead mice sample. So something was there which made the R strain transform into S strain. It was done somehow. So R strain bacteria somehow being transformed by heat killed S strain bacteria. So some transforming principle transferred from heat killed S strain enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. So this must be due to transfer of genetic material. But this also did not clear what was the nature of genetic material. This was the basic understanding of Griffiths experiment. In our coming video, we'll be talking about biochemical characterization. Whether scientists were able to prove that out of protein, RNA, and DNA, what was doing this transformation. Keep watching, keep solving questions, and also keep reading textbook. That will help you to do well in your examination. Meet in the next video. Till then, keep reading.